It's great to have this turnout, and this is really one of the college's signature events, and certainly one of the most enjoyable. I say that because we really get to celebrate extraordinary research accomplishments of two members of our faculty, and celebrate it in a way that I think at this really hectic time of the semester we can just pause and celebrate their success, as well as get to really immerse ourselves and learn about some of that outstanding science. Before we get to the DeLisi Award and lecture, it's my privilege to introduce the winner of this year's Early Career Research Excellence Award for the College of Engineering. This year's winner is Hadi Nia, Professor Hadi Nia. I think he's here. He can stand. Yes. Okay. All right. Okay, so Hadi joined us in 2019, and he has built a high-impact research program that combines principles of engineering and physical sciences with molecular biology to answer key questions in cancer and pulmonary pathophysiology. He has also published groundbreaking work on quantifying the physical abnormalities in the tumor microenvironment, and he has been at the forefront of research in developing technologies for visualizing lung pathophysiology in a functioning lung in real time and at the cellular through whole organ scales. Hadi has received the NIH Director's New Innovator Award, which is given to only 72 awardees in the entire NIH community, an NSF Career Award, and he is the first Boston University faculty member ever to receive a Beckman Foundation Young Investigator Award while here at BU. He's also received an NIBIB Trailblazer Award, among others. His work has received over 2,600 citations, and he's also excelled as an educator, bringing new in-class demos and examples to teach undergraduate and K-12 students transport phenomena and fluid mechanics. Please join me again in congratulating Hadi. Okay. And now I'll turn it over to Ken to introduce the DeLisi Award.
Well, thanks, Elise, and thanks, everyone, for your participation and your taking the time out to come to our signature event. First, let's start by thanking the extraordinary individual which made this event a reality, Charles DeLisi. I introduce Charles every year, but there's always new people, so I get to say, you'll hear it again, but they'll hear it. Some of the new people hear it. Charles is a transformational figure in the world and a transformational figure at Boston University. In the world, he is widely known as being the phrase, the father of the Genome Project, the individual at the Department of Energy that got the government convinced to invest and to sequence the entire human genome. And as a consequence of that, obviously, medicine has changed for all time. He became the dean in 1990 and was transformational in creating one of the most exciting colleges of engineering in the nation across the board and put BU's College of Engineering on the map. And we're deeply indebted to Charles, not only for those two things, but for his generosity and graciousness in endowing this lecture series. So please join me in a round of applause to thank Charles DeLisi for his impact on the world. So, we are honored to announce that the winner of the DeLisi Award in 2020-23 is David Boas. The award celebrates a distinguished scholar in our community who has made outstanding contributions to engineering and to society. David is the Arthur G.B. Metcalf Professor in Science and Engineering. He received his BS and his PhD from RPI, from where? Okay, PhD UPenn. Forget RPI. He's widely recognized as a pioneer in the development of novel optical imaging methodologies for the applications in neuroscience, neurology, and breast cancer management. He led the development of one of the first imaging systems using continuous wave near-infrared spectroscopy, NIRS, for functional imaging. These systems are now being distributed commercially to 5 continents. David's group was the first to introduce laser speckle contrast imaging to the neurosciences for real-time imaging of cortical blood flow, and he led the first in vivo imaging of spatiotemporal profiles of oxygen partial pressure in tissues. In the domain of breast cancer management, David has integrated optical breast imaging for simultaneous imaging of breast structure and function, analogous to the combination of PET imaging and X-ray computed tomography, to improve image sensitivity and specificity. He is the founding editor of the Journal of Neurophotonics, and in 2017, he launched the BU Neurophotonics Center, which now has 50 BU faculty members from 8 departments in different schools and campuses, and many more students and postdocs, of course. He is a fellow of the Optical Society of America, SPE, and the American Institute of Medical and Biological Engineering. He is also the recipient of the Britton Chance Award for Biomedical Optics. His work has been cited over 51,000 times. So Hadi, a few years later, you got to get over there. His H index is 122. He's trained 30 postdoctoral fellows, many of whom are now in faculty positions. He also has supervised, or currently is supervising, 27 PhD students. Please join me in welcoming David Boas to give his DeLisi lecture on illuminating the functioning brain to reveal the role of blood flow.
All right, thank you very much, Ken, for that wonderful introduction. It's a great pleasure for me to, of course, receive this great honor, and also to have so many people here present for this presentation. Very happy to see all of you. You know, many, if probably most of you from BU, but also I'm very happy to see many of my former colleagues from Mass General Hospital here as well. So today I thought I'd tell you a little bit about the story of how I got here, with some science along the way, kind of all related to kind of the major theme of my own work has been, you know, using light to study blood flow in the brain and how that's relevant for us. My path, I started off as an undergraduate at RPI. I have no idea where Ken got this idea. But I was very much interested in electrical engineering, but also skiing. I grew up in South Jersey, not much skiing there. I needed to go to a college that had more skiing, so I was attracted to RPI for more than one reason. But I quickly realized I wanted a more foundational education, and I switched to physics. I devoured my physics textbooks. I really loved it. And I've gone back and I found 8 pages that talk about the Doppler effect. I love all the physics, but in particular, this was important to me because so much of my professional academic career is based on the Doppler effect, as you'll see. Many other things happened. I won't have time to tell you about all the stories in college, but you know, at the end of college, I was faced with, should I be a ski bum or should I go to grad school? And I realized I was as passionate about physics as I was about skiing, so I thought, let me try grad school. Physicists in grad school were not getting any jobs. I figured I could always become a ski bum after grad school. So I went to University of Pennsylvania, and during the first year seminar, I met Arjun Yode. He gave a lecture on colloidal systems and how he used diffusing light to study the colloidal systems. He was a soft condensed matter physicist, and he was studying the formation of colloidal crystals using diffusing light. And I was very interested in that, so I went and talked to him. And he said he had just met this professor in the medical school, and he and the other professor were thinking of getting a joint PhD student. He suggested I go meet this professor, so I took a random walk, as we like to do in the field, to Britton Chance's laboratory, and I met him. Turned out he's a pretty famous man, so I need to tell you about his background. He started his PhD in the 30s, studying enzyme reaction kinetics using optical spectroscopy and stop flow device. He was interrupted during the war, where he was a radio guy. He was at the MIT radar lab, made contributions there, but then he went back and fully detailed the process of oxidative phosphorylation. He's well known for that. He always expected, and many people expected him to get the Nobel Prize for that, which he didn't. He has good stories why. But he did, in the 1952 Olympics, get the gold medal for sailing. So at least he has that. And you know, during his summers, he would invite his lab down to his place in New Jersey and have us clean the bottom of his boat. And the name of this boat was Complex 4, and I thought, Britton Chance, he's a complex guy. But actually, it was named for complex 4 in the electron transport chain. It took me years to figure that out. He actually won the gold medal in complex 2, right? So it all fits together. He went on after figuring out this energy metabolism at the electron transport level. He wanted to study energy metabolism in humans. He did the first MR spectroscopy of intact organs in humans. He wanted to develop optical methods to complement that. Got into near-infrared spectroscopy. This was the launch of biomedical optics. And this is kind of when I showed up in the lab. So it was a really great time to walk into his lab. And so I joined the Chance and Yode efforts to use photon propagation through highly scattering medium to study tissue absorption, scattering, and dynamics. We did lots of fun experiments, a fellow grad student and I, studying these propagation of diffuse photon density waves through highly scattering media, imaging, absorbing targets there, trying to convince ourselves that that was like imaging a breast tumor or imaging brain function, which we then went on and did many years later. But a lot of what I did was actually working with long coherence length lasers in Arjun's physics lab because we wanted to study the dynamic properties, and that you need a coherent laser. So I developed a theory that would describe how correlations of speckle intensities would diffuse through highly scattering media, and we could form images of the spatial variations in the dynamic properties of the medium, just like we could the optical properties of the medium. We did the first human measurements of tissue dynamics through thick tissues using this laser system. And this is a normal near-infrared spectroscopy. So I'm contrasting near-infrared spectroscopy with this diffuse correlation spectroscopy. Near-infrared spectroscopy measures the absorption of hemoglobin in tissue. And so using different wavelengths of light, we can measure oxy and deoxyhemoglobin. And a classic experiment is you occlude blood flow in your arm, and basically there's no more blood flow, so all the oxygen in the hemoglobin is depleted. And so you see that when we raise the pressure, deoxygenated hemoglobin increases, but the total hemoglobin stays constant when we raise the pressure. We also had this measurement of blood flow, the dynamics of red blood cells, and we see that blood flow drops down to 0. And as we slowly release the pressure, everything returns back to baseline, but first going through hyperemia. I never heard that word, but that turned out to be a very important word again in my career because hyperemia is involved in the functional hyperemic response to neural activity, which is kind of the basis of the bold fMRI signal and the functional near-infrared spectroscopy signal that I'm going to be telling you about for measuring human brain function. One curious thing, though, we were measuring the correlation function of the intensity fluctuations of laser speckles, and the theory I had derived said that should decay exponentially, linearly with time, proportional to the velocity of the red blood cells. Oddly, though, it was decaying as the square root of time. I wanted to graduate. I couldn't figure that out, and I graduated anyways, not knowing why it looked like diffusive motion. And so culminating in my PhD, it was an amazing lab to work with. Many trainees were in the lab when I was leaving. I think many of you recognize many of these names. They have big labs of their own around the world. But I also met many other senior faculty mentors who were just amazing people. Marco Ferrari did the first clinical near-infrared spectroscopy measurements in infants in the 80s. David Delpy, a physicist from London, he developed the mathematical framework for near-infrared spectroscopy. And then Arno Willinger, he did the first functional near-infrared spectroscopy measurements of human brain function. So able to meet all those people and many more. And oddly, I found myself without a job. I thought I had a job, but then suddenly I graduated and I didn't have a job. Britton Chance said, "Don't worry, David, do nothing. Wait 2 weeks, don't be rash." 2 weeks later, I was at a conference. I met Paul Kelly from Tufts University, and he said, "We have a job." So he hired me as a research assistant professor at Tufts University. There they had an electro-optics technology center, which was serving this rapidly growing photonics industry in the Boston community and world. And we were training lots of industry people and masters. It was a lot of fun. But then the very next year, BU opened a much, much larger photonics center. So we were a little bit nervous about that. So I was sent over as a spy to check out my neighbors. And I attended the inaugural opening event, the Photonics Symposium. But actually, independent of that, I was already connected with BU medical school faculty trying to apply near-infrared spectroscopy to clinical situations. We published a paper. We were interested in trying to commercialize the technology. Photonics was looking to incubate new companies. So we actually tried to start a company with BU Photonics Center in 1997. So we needed patents from Britton Chance. So actually, BU paid me as a young research assistant professor to fly down to Philadelphia, visit my old mentor, but he had different ideas for me. So he and Arno and Villinger, they were rapidly developing functional near-infrared spectroscopy to measure human brain function. So these were Arno's first measurements. Arno, just a few years before that, had worked with Bruce Rosen. So he was well aware of when Bruce Rosen published the first fMRI measurements of human brain function. And so Arno, very ambitious, he was very interested in methods for measuring human brain function. He organized a meeting in Berlin where he works, and he brought Britton Chance and Bruce Rosen. That was the first time Bruce Rosen from MGH had heard about optical methods and how it could be used to measure human brain function. A few years later, he found some money to hire an optics person, and Chance and Villinger pointed to me. In parallel with that, I was on soft money at Tufts University. I was talking to clinicians all over Boston trying to figure out how to use near-infrared spectroscopy. I met Walter Koroshetz, who was director of the stroke unit at MGH, and I got my first grant, $20,000 from CIMMYT, had just started at MGH, and we built this first FNIRS imaging system. David Benaron, who was a physician at Stanford at the time, he saw that system. He said, you got to come visit. We'll measure my premature infants. And we did that. We took the system out, got one image every 5 seconds, but we were able to show brain activation on these premature infants. So super exciting. And MGH was impressed, and so they offered me a job. So I went to MGH, and that was a crazy exciting time. There was only 40 papers in the field of FNIRS for human functional brain imaging. And just 5 years before that, the first fMRI experiments had been done in humans. So there was a lot of activity trying to understand this hemodynamic response to neural activity and how you can learn about human brain function using that. And they really wanted optical methods to help them better understand the MRI signal. So we arrived at MGH, and we had to build a better system. That system I showed you was getting an image every 5 seconds. We needed much better temporal resolution. Fortunately, I had Andy Siegel I was working with, and he was a really good electrical engineer, and he built this prototype system, and we measured basically changes in hemoglobin concentration every time he tapped his fingers. So when he taps his fingers, the motor cortex and the contralateral hemisphere has increased neural activity. There's increased blood flow, delivering more hemoglobin, which absorbs more light. And so we see this decrease in the intensity of light we detect non-invasively on the scalp. And every time he rests, it goes back, and taps, it goes down. So great. Then Winfield Hill visited us. So probably many of you have read this book or know this Bible of electronics. He said we were doing great electronics. Only recommendation he had for us was to pay a contract engineering company. He introduced us to TechN in Hour West, and we went through several iterations of systems with them. With this fourth and fifth generation system, I think we published 100 papers with these systems and actually sold over 100 of these systems around the world. So that was a great success. But we also had to do science with it. So kind of our first task was to better understand the fMRI signal, the blood oxygen level dependent, the bold signal, which was basically representing deoxyhemoglobin. Deoxyhemoglobin is paramagnetic. It perturbs magnetic field. Oxyhemoglobin is diamagnetic. And so this gives rise to perturbations. You know, when you have neural activity, it changes blood flow, it changes deoxyhemoglobin, it perturbs the magnetic field, and you can pick that up in an MRI image. We were measuring oxy and deoxyhemoglobin using absorption spectroscopy, and so we could do the two simultaneously and look at spatial temporal correlations. And so we did that, found very good spatial temporal correlations. This is just picking the signals from that ROI. 2-second finger tapping task. The hemodynamic response is always slow, right? Neural activity is much slower, but a blood flow response is slow. We see oxyhemoglobin increasing, total hemoglobin increasing, blood flow is increasing, pushing out deoxyhemoglobin, it decreases. Less perturbation of the magnetic field, and so that, as a result, the bold signal goes up. Okay, so that's great. So we wanted to do more with that, so some physiological modeling. The inputs basically are arterial dilation and oxygen consumption. There's neural activity, but the neural activity has an increase in oxygen consumption, and it releases vasoactive agents that dilate the arterioles. So building a biophysical model, we could fit that data and get the input functions, which allowed us to estimate the cerebral metabolic rate of oxygen, right, with very good spatial temporal resolution. So it's quite interesting. And there's many things interesting about it, but I'll just point out this flow consumption ratio. So blood flow pretty much always increases 2 to 3 times more than the oxygen consumption response. So people were asked for over a decade, why is that? It's pretty simple, but you have to debate it for a long time. Basically, you need a steeper oxygen gradient from the capillaries to the mitochondria to support higher oxygen consumption. The brain does not have a reserve of oxygen, and so the only way you can get higher oxygen metabolism is to get a steeper gradient. The mitochondria is already pretty close to zero, so the only way you can do that is raise capillaries very high, and you only can do that by raising blood flow a lot. Okay, so at this point, our hardware was way ahead of analysis and experiments. So we spent a long time developing analysis methods, software packages that were used by thousands of people still today. We then ran an educational workshop for 20 years every year to bring in more users to the community. After about 10 years of doing that, we realized there needed to be a conference to allow the early adopter users to present their work to other early adopters and meet technology people. So we ran a conference. It was a big success. We did that at Harvard. And so we ran it in London. It was an even bigger success. And then Montreal, Paris, we went to Tokyo. And then last October, we came back to BU. We formed a society along the way, wonderful community. If you have any interest, join the society, come to our conferences. Where's the next one? Next one's in UK, Birmingham, in 2024. Okay. So that was the human measurements, but there we were at Mass General Hospital, and we had all these translational neuroscientists who wanted to study the brain in animal models. And so they were asking us, well, could you measure blood flow? Could you measure oxygen consumption? And so we developed the first multispectral imaging system and laser spectral contrast imaging system, which I'll tell you about. And we also started utilizing 2-photon microscopy and optical coherence tomography, which I'll tell you a little bit about. So first, multispectral reflectance spectroscopy is essentially a lot like FNIRS. We're doing absorption spectroscopy to measure hemoglobin concentrations, and this laser spectral contrast imaging is like this diffuse correlation spectroscopy. We're measuring speckle intensity fluctuations of laser light, Doppler shifts to measure blood flow. And so this was a rodent, and we were doing a 4-pulse stimulation and looking at the motor sensory cortex, and we could see these localized changes in oxyhemoglobin, deoxyhemoglobin, and blood flow. Look at the ROI, see the time traces, looks a lot like in a human. Flow consumption ratio is about 2.5, but what's nice about the animals, of course, is we can get much more invasive and study that in much more detail. And really, right around that time is early 2000s. Anna DeVore showed it at MGH. She wanted to study neurovascular coupling, but with using MRI. And for my great fortune, the MRI machine broke when she arrived, and she couldn't study neurovascular coupling using MRI. So she decided to use optical methods. This was a 4-pulse stimulation again. This is what we did often. And you could see this increase in total hemoglobin concentration in the contralateral cortex and a decrease in total hemoglobin concentration in the ipsilateral cortex. And so conventional wisdom was that basically blood flow was increasing to support increased metabolic demand of neural activity. So if we saw if we saw blood flow or blood volume decreasing, that would suggest there was reduced metabolism and reduced neural activity. But Anne, as a neuroscientist, she knew quite well that actually neural activity was not reducing here, it was increasing. So this was very curious. There's a disassociation, but everyone says blood flow increases to support metabolic demand and neural activity. That's not what's happening. So she went in and did voltage-sensitive dye imaging, confirmed, sure enough, there is depolarization of the neurons, but you can actually see in this ipsilateral cortex, there's actually relatively more hyperpolarization after the depolarization. So this led to a very simple model that the depolarization of the neurons releases vasodilatory agents from the synapses that causes the arterioles to dilate, and the hyperpolarization caused by inhibitory cells was basically releasing vasoconstrictive agents causing the arterioles to constrict. Very simple model. She did lots of experiments, confirmed that that model was true. The curious thing was that, here we are in this presence where we have increased neural activity. There should be more metabolism, but flow is decreasing, right? So she needed to confirm that metabolism had actually increased, which she did 2D autoradiography, showed that glucose metabolism had actually increased. So this was very important because it demonstrated blood flow is not responding to oxygen demand like everyone had thought, but it's actually anticipating oxygen demand. So what would happen actually, if you were to continue this stimulus for a minute, that inhibitory activity in the ipsilateral cortex would actually suppress neural activity, right? And metabolic needs would go down, right? So, but by fooling the system this way, you could kind of tease it out, what's really going on. So this then led to the question, what is the impact of such flow metabolism changes on oxygenation at the microscopic level? So we started doing 2-photon microscopy to measure oxygenation in individual capillaries. So this is a lot like FNIRS in that we're measuring oxygenation, but it's not, because we're not doing absorption spectroscopy. We're actually measuring phosphorescence lifetime. So a colleague, Sergei Vinogradov at University of Pennsylvania, amazing photochemist, developed this phosphorescent agent that's quenched by oxygen. And the lifetime is directly inversely proportional to the partial pressure of oxygen, and you can calibrate this. So we could put this dye into the bloodstream, and we can measure phosphorescence, and we could go to individual vessels and measure phosphorescence lifetime and quantify partial pressure of oxygen. We were also adopting optical coherence tomography at that time, which is a lot like Doppler. Well, it is a Doppler method. We're basically using Doppler methods to measure blood flow, but with a resolution at the capillary level. So I'll tell you a short story about each. So measuring oxygen, Sava Sakadzik, he measured oxygenation in arterioles, capillaries, venules in many different mice. So this is a collection of 6 mice. And aggregating the results could look at the oxygen saturation. You can convert between partial pressure of oxygen and hemoglobin oxygenation and get the hemoglobin oxygenation versus arterial diameter. And so the first thing you take from this graph is that arterioles are delivering 50% of the oxygen to the tissue. Right? So the veins, that's the oxygen leaving the brain. This is the oxygen entering the brain. Half of the oxygen has already left the blood before you reach the capillaries. You go to any textbook, it says oxygen is delivered to the tissue from capillaries, right? So clearly that's not the case. People knew that, but it's too hard to describe this in textbooks, so you just say capillaries deliver oxygen to the tissue. And it makes sense because capillaries, at least in the brain, are only like 10, 20 microns away from any cell, or I should say any cell is no more than 20 microns from any capillary, right? But in arterial, there's, arterioles penetrating into the brain are separated by 400 microns or so. So it doesn't make sense that so much oxygen would be delivered to the brain from arterioles. Just doesn't make sense. So the other interesting finding was that looking at the capillary oxygenation, the vast majority of capillaries are below the venous oxygenation. So this was very confusing to us. The veins, the capillaries drain into the veins. How could they have less oxygen than the veins? Very confusing. Well, it turned out it's pretty easy to explain because basically what we're measuring in the capillaries is a spatial average, spatially weighted average, and what we measure in the vein is a flow-weighted average. So basically, there's a small number of capillaries with high flow and high oxygenation because there's no time for the oxygen to diffuse into the tissue. But there's many more capillaries with low flow that deliver all of their oxygen to the tissue. And the veins are biased by the high flow capillaries. So that's basically what's going on. so there must be a reason why there's a few high flow capillaries. We have ideas that basically in times of increased metabolic demand, you can kind of squeeze those capillaries and all of that oxygen goes out to the rest of the capillary network. We know how to do those experiments. They're complex experiments. We haven't done them yet. We want to do them. But the data we have so far, we are able to show this new mechanism of oxygen reserve in the brain. I'm not showing the data here, but basically when you increase blood flow, you suddenly go from delivering 50% of the oxygen from the arterioles to delivering like 10% of the oxygen to the tissue from the arterioles. So you're shifting, almost twice as much oxygen into the capillaries where it can be delivered to the cells, right? So that was a novel finding from these data that we were able to extract. Now, final story, the OCT that I want to tell you about is we did many things with OCT, but one thing I was proud of was we were able to go back to my PhD and explain 20 years later why RBCs look like they're behaving like diffusing particles. So we were able with OCT to zoom in on individual penetrating arterioles and measure, because we're doing a correlation function, we can distinguish flow from diffusion particles, right? And we can measure laminar profiles of the flow in this penetrating arteriole, and we can measure diffusion. And basically what we're seeing is red blood cells look like they were diffusing near the vessel wall. And so we looked in the old literature, and basically there's old literature, 70s, describing how the shear of blood flow produces a tumbling of the red blood cells that looks like diffusion. And it turns out that this diffusion actually dominates the speckled decorrelation. Right, so finally measured this in dozens of arterioles and was finally able to kind of explain what we couldn't explain 20 years earlier. Okay, so that was fun. Okay, so it was 2015. We were doing lots of great stuff at MGH, worked with many great people in the optics division of the Martino Center. We were developing novel optical methods to study the brain and other organs. We were working with collaborators who were interested in clinical problems and translationally relevant problems. But I started to think about moving to a university and working with students, getting back to engineering and the basic sciences. October 2015, I got a call from Irving Biggio, who asked me, if I knew about this search that they were doing in BME. And I said, that sounds interesting. And then Jerome Mertz and Tom called me up and invited me to dinner and introduced me to Tom Buffano. They said nothing about the job search, but now I suspect otherwise. And then in November, Mike Hazelmo, I met him at Society for Neuroscience. And so that really got me thinking about growing the neurophotonics cycle, of going beyond translational applications for photonic methods in the neurosciences, but we could grow that to basic neurosciences as well and work with many more faculty than we had at MGH. So I was excited about that. And so I applied. I got to come on my birthday to give a job talk, which I thought was a smart thing to do. Everyone was nice to me when I told them it was my birthday, you see, so I highly recommend that, right? Coincidentally, a fun fact, Chantal Stern interviewed me at BU. She also interviewed me 18 years earlier at MGH, where she was Bruce Rosen's, I believe, first postdoc and the first cognitive scientist to do fMRI experiments, right? So she has interesting background as well. Now, 10 days later, I was given the Britton Chance Award, so you know how much I respect Britton Chance, so it was a great honor for me to receive this award. I was not thinking about the BU job, but then I was invited back several times to visit BU, and actually I got to, one of the days I came back to meet with people was actually the 2016 Charles DeLisi lecture. And so I got to attend that lecture, enjoyed that lecture, and enjoyed the reception afterwards, which I hope all of you will do as well. So then in June, I remember I got an offer from Ken, and I was concerned about just leaving a great community at MGH, but of course I'd be joining a great community at BU, but I had to think about it, so I do what I tell all my mentees to do, take a hike. So I hiked up Mount Washington before I responded to Ken. It was a beautiful day. This is Tuckerman's Ravine. It's not often that beautiful. And Lake of the Clouds Hut, you can see Mount Washington. It's like one of the few days, cloudless skies, one small cloud. walking up to Mount Washington, looking back at Lake of the Clouds Hut, looking over the Presidential Range to the north. Of course, all this time I'm thinking, what should I do with my life? And then got to the summit, and this is what I always tell my mentees, you get to the top, you know what you want to do, so yeah, I'm going to BU, but I'm also gonna not leave MGH. It's only 4 miles away to Charlestown. It's easy for us to keep working together, and we can make both communities stronger, right? So I've been working on that. I think it's been working. So July 2017 started at BU. The lab was being built. That was very exciting to be part of that. So couldn't start in the lab, but could move into my office, showed off the nice view to my son, and then started planning the 21st annual photonics symposium on neurophotonics. And it was 20 years, 1 month, and 1 week since the first photonics symposium that I had the pleasure of attending. And never did I imagine that I'd be running my own photonics symposium, let alone being a professor at BU, or for that matter, directing the Neurophotonics Center at BU. And so with that symposium, we launched, opened the neurophotonics space. A few months later, we had a symposium for FNIRS, generating a collaboration of more than 10 faculty on campus and all of their trainees. And then over the last 5 years, we're very successful bringing in lots of faculty and starting new projects. And last summer, we were able to renew the Neurophotonics Center, which brings us to where we are today. So we have a great team of associate directors and technical directors and staff and project scientists who are able to work with and facilitate new interactions and collaborations amongst the 50 faculty who are mostly pretty actively engaged in the center, working with many different technologies. This is all really kind of brought together by the community of students that have come from 8 different departments. And it's really kind of them who are making the community much stronger, as evidenced by this sociogram, where we see that there's just much more intermingling of neurosciences scientists with photonics scientists through this community. So also BU has been fantastic at hiring a bunch of new junior faculty in the neurosciences and in optics. And so we've been able to work with them and support them in adopting and developing and adopting novel technologies to study the brain. They're obtaining new grants and taking on leadership positions, presenting their work at conferences, and really kind of raising the reputation of BU as a place to be for neurophotonics research. So in the last couple slides, I just want to tell you about some things going forward that I'm very excited about. So one of them continues to be FNIRS. FNIRS, the first experiments were done 30 years ago now. I've been working in the field 25 years, yet there's still many interesting things to do. So one is just the miniaturization of electronics has made it much easier now to make wearable FNIRS systems. So we got this new Brain Initiative grant working with our team, and we're building the hardware now to enable us to make these wearable systems, and working with the 10 faculty at BU who want to apply that technology. And so what we want to do with it is measure perception, interaction, walking, which are behaviors, human behaviors, that are rather challenging for us to measure with fMRI. But we can, with wearable technologies, in particular FNIRS and FNIRS combined with EEG, we can measure these behaviors in ecologically valid settings, right? And these are complementary because FNIRS is measuring the hemodynamic response to neural activity, and EEG is measuring that electrical response arising from the neural activity. So there's still lots to explore in how we can integrate those two signals, particularly if they're acquired simultaneously. People have been talking about this for over 2 decades, but still not much has been done there because combining EEG with fMRI, not very easy, right? But EEG with FNIRS is much easier. So that will be fun. And we're working now with all the cognitive scientists trying to figure out what are the cool experiments we can do when we remove all of those constraints that you have with traditional functional neuroimaging studies. So this is the status of the system. last week. This was our prototype headgear. So what's cool about this is that it's going to be high density FNIRS measurements. So it gives us about twofold better spatial resolution than traditional FNIRS measurements. This is the system as it exists now. It was built by Bernard Zimmerman doing all the electronics, and Joe O'Brien has been doing all the mechanical design work to enclose everything. And the system just functions remarkably well. We're very happy with it. We were thinking all along that this would be in a backpack, and I would be wearing it. So for the next few months, I'm going to be testing this out, figuring everything out, but I'm sitting at my chair all the time. And I realized I can't wear this on my back if I'm sitting at my chair. So Chantal Stern told me, get a baby carrier. So I got a baby carrier. And so now I can wear my new baby in my chest and measure my brain activity at the same time. And this is working very well. We right now are populated the frontal regions, but in May we will fully populate it with a high density array of optodes. And so what we're going to do with that is measure brain activity during walking, and in particular how the brain function is disrupted in Parkinson's disease and maybe in survivors of stroke who have trouble walking. We're going to be looking at people interacting, conversing, and in particular looking at language impairments post-stroke and how you can optimize therapies for those patients. We're going to be looking at perception and attentional errors or working memory errors that can happen in neurodegenerative diseases. We're not, so we are starting some of those studies, but for the next few months, I'll be wearing this device, figuring out the ergonomics of it, and I'll be talking to colleagues in my office while wearing this device. I'll be writing grant proposals, reading papers. I'll be walking to meetings, right? So if you see me, you know what I'm doing. This was 2 weeks ago, wearing the device for the first time, and I was quite excited. I walked all over the Silsey building showing everybody that there's clean signal, no motion artifacts. Okay. But of course, our colleagues, at BU are running a lot of experiments with the previous generation system, which was portable, less wearable. So this is Lou Awad and Regina Slutsky, who are using a low-density FNIR system, only looking at the frontal regions. They're looking at stroke survivors who have trouble walking, have asymmetry in their propulsion, and they help them improve mechanical efficiency in their walking. But the question has always been, well, what is happening to their cognitive efficiency? Because they have to think about every ankle flex as they walk. And so they can't think about anything else while they're walking. And so they've been using FNIRS to measure the cognitive load of walking and how their exosuit can reduce that cognitive load. And so they're getting really exciting results. Now, of course, in addition to them, there's many other colleagues with many other exciting results. And I really have to give a shout out to Miriam Uchel, who's working with all of them. I don't know how she does it because they have large teams, each of them, and they're all getting some pretty exciting results. Finally, I grabbed my group yesterday. We got a nice photo. It was a beautiful day yesterday. I want to just shout out to them. You've heard about all of the FNIRS measurements we're doing. You've heard me talk a little bit about speckle contrast. We're still doing a lot of interesting things with laser speckle contrast, but I'm also interested in ultrasound speckle. And so we've been doing functional ultrasound measurements of speckle dynamics. We still study neurovascular physiology. We got 2 new grants looking at stroke and Alzheimer's disease. And I'm very happy that Anna DeVore somehow ended up at BU as well. And we continue to collaborate on this. And also working on volumetric histology with Irving Biggio and Ann McKee. So we have a large group of people working on that. And finally, with that, I'll just thank you for your attention. I'd love to have some discussion.
That was fantastic, David. We have time for several questions. We have microphones, 2 people with the microphone. So if you want to ask a question, wait until the microphone gets to you, and then you'll ask the question. So questions from the audience. I'm going to start with a question. So let's talk about the stroke rehabilitation.
Yeah.
So when we walk, everything we do is subconscious. We don't
Right.
think about
Right.
what's going on.
Right.
And how will this device kind of get a sense of, are we redeveloping the neural circuitries that we need to not impact the consciousness, or the role that consciousness plays now in just the walking for stroke patients?
Right. I mean, so they can talk. Lou and Regina can talk an hour about that topic. I'll try to give you just a couple minutes from what I've learned from them. So physical therapy, they measure biomechanics. So they work on efficiency of propulsion, of walking propulsion. But of course, their patients always talk about they can't think about anything else. With this device, they've been able to show, so they have this exoskeleton which can do the work of lifting the ankle as they walk, but there's a timing of when you lift. And that affects the walking efficiency, propulsion efficiency. And they look at how that also changes cognitive demand. And so what they're seeing is that actually you can optimize for walking mechanical efficiency or cognitive efficiency. Right, and every patient has a different profile, and so depending on your physical therapy regimen, you will tune it differently for every patient. So maybe initially you want them to have cognitive load to walk more efficiently, so they can learn how to walk more efficiently. But maybe there's people who are maybe never going to get better at walking, and so you accept mechanical inefficiency just so that they have cognitive efficiency, right? So it's going to be case by case per patient. Right? Yeah.
Microphone for John? I can talk. There's two Johns.
I have the mic. I get to go first. Hi, David. Thank you. Great talk. I found myself thinking, how far could you push this, and could you really take a dent out of the need for fMRI? So in the limit of maybe thousands of sensors and thousands of emitters and maybe a three-dimensional model of the brain from structural MRI, how far could you push it? And what would not ever work, no matter what you did?
Right. I kind of remember. Well, I won't go there. But I hear many people ask MRI one versus the other, right? But it's never going to be one or the other. They complement each other. Right? So fMRI has amazing spatial resolution, the entire brain. With FNIRS, we're only measuring the brain close to the skull, but we can do that in a wearable system, and we're not constrained by the patients or subjects we can measure, right? So they're just highly complementary. But as we push the technology, we can probably in adult humans get a centimeter spatial resolution on the brain close to the skull, right? And we should be able to do that like in a baseball cap device, right? We should be really able to make a truly wearable system.
And how
right.
far down?
Oh, yeah, 5-10 millimeters. Yeah, not very deep, right? But fortunately, a lot of the brain, functionally relevant brain, is accessible. And those functionally relevant regions that are not accessible project to networks that are accessible. Right? So lots of utility as a result. Right?
Okay, so is this working? Okay, so that was exciting to me, but I'm going to ask a totally naive question because I don't understand most of the details. But I'm very interested in the way that brain networks evolve from birth to end of life. And, you talk about, to follow up on Ken's question, we are stabilized inverted pendula, when we stand up. But I don't believe that we learn to stabilize by doing various types of analysis that we do in my control systems course. It's really sort of reinforcement learning, and the brain evolves, and somehow after a while, everything starts to work, and then at the other end, function starts to deteriorate, and we become somewhat unstable. Are those evolutionary processes interesting to study?
Absolutely, and I think this is the type of technology you need, as it will enable you to do longitudinal studies, not just cross-sectional studies, right? So people are trying to study that now, and challenged because of the cross-sectional nature. And of course, also you're stuffing people in the tube of a magnet, and so you can't capture natural behaviors. But with this, we can capture the natural behaviors. I'm excited working with the colleagues here. We're working on the older end of the spectrum with neurodegeneration and such. It'd be very exciting to do it at the developmental end as well. And we have done studies in infants, and up to 12 months of age, they're pretty good subjects. After 12 months to like 3, you know, there's the terrible 2s. They're really good at ripping things off their head. And you can't really reason with them. But people will figure that out. Right.
Other questions? All right, next up.
Since I'm right here, I'll ask an easy one. Maybe it's easy. In your wonderful new system, are you still limited by instrumentation bandwidth and dynamic range? Or has that caught up and the limitation is the human body?
It was fascinating to me. We were using expensive, sensitive avalanche photodiodes, photomultiplier tubes, and the wearable systems were just using pin photodiodes. And it works amazingly well. It's crazy. Of course, we do want better sensitivity. There's these new silicon photomultipliers. We want to adopt them. That will give us basically a almost 20-fold improvement in the noise floor, which we don't really need. However, it helps us with hair, right? So actually for like just making it more accessible routinely to all hair types, that detector is going to be very important.
But the basic, the bandwidth is kind of there.
Bandwidth is there.
Yeah.
I want to ask a quick question.
Battery efficiency is something we can improve on. But
Alzheimer's disease, 2 of the biggest challenges is early detection and then whether therapies are working or not. Can you speak a little bit about, you hear a little bit about people now, their gait, their way they speak is an early perhaps sign of emerging. Would this system, or has this system been used to kind of correlate that, yes, we're picking up functional changes in the brain that are consistent with that? And on the back end, how could this be used to say, I'm going to give this drug or this therapy? How do you know if it's working or not?
I mean, so we tried over 10 years ago now, working with a pharmaceutical who was very interested in early detection because a lot of their trials fail because they don't identify the patients early enough. And so we have a whole protocol written out, and we should run that protocol. Turns out just AD patients are highly subscribed to research studies. So we're working on figuring out how to run that study. But absolutely, I'm just super excited by the work of Yannis Paskalidis, Rhoda O, and others at BU, where they're just using digital recordings and as early biomarkers, and they're able to diagnose onset of dementia. I think a device like this will be a great complement to that.
Time for a couple more questions, if any, in the audience.
There's a question here.
You talked a little bit about combining signals from what's called EEG, electrical signal, and the blood flow information. I'm wondering what kind of task where these 2 signals will like explain what's going on in different ways. Do you have like specific examples?
I wish I was more versed in EEG. We're working on it. So EEG measures evoked responses, 200 milliseconds time scale. That will be highly complementary to our several seconds hemodynamics, but EEG also has rhythms, alpha rhythms, alpha, beta, gamma, and looking at modulation of those rhythms, I think will also be very interesting in how they compare to the hemodynamics. Specific questions, ask us in 2 years, right, of how we combine it. Right?
I will, thanks.
Well, I think I want to thank David one more time. Before we, okay, Ted, thank you. Now, David was just named the Metcalf Chair, and if we knew that was going to happen, we wouldn't have had this one made up. But he can keep it, a special chair that honors him.
Thank you.
And a framed version of the announcement of David's winning of the Charles DeLisi Award. So we get